welcome. We have been discussing spike train distance based quantification of dependence between stimulus and response. So, just to recall, we have a set of capital N stimuli, and based on the spike trains produced by each of those stimuli and each of those repeated multiple number of times, let us say capital M times, as in the past lectures, we compare spike trains or compute distances between spike trains, and then assign each of the spike trains to a particular stimulus to which its average distance is closest to, and based on that we get a confusion matrix, and from that confusion matrix can be treated as a joint distribution between the stimulus and the estimated stimulus based on the spike train, and from that joint distribution we quantify the dependence between the stimulus and response. Of course, it is a lower bound as we have discussed earlier. So, the main thing there that we had set aside is the choice of the discrimination metric, that is how the spike train distances are computed. So, we will begin by introducing the ideas behind choosing spike train distances based on what our question is. So, let us go back to the problem, and let us say we have let us say stimulus 1 and stimulus 2. We have two spikes in response to the stimuli. Let us say for spike train 1 it is at this particular time, and for spike train 2 it is at this particular time, kind of the example that we had talked about in the very beginning of these discussions. So, now the question is, in some ways that given these two spikes, we will be able to say that they came from two different stimuli most likely. That is only when we have a particular temporal resolution at work for the postsynaptic neuron. So, it will depend on how precise or how precisely the postsynaptic neuron integrates information over time, that is, what is the resolution at which it changes its responses over time. So, let us remind ourselves what a neuron model was from our initial third of the course, where we have a capacitor and then the ion channel based branches with a battery. So, let us say this is a sodium branch, and then we have a potassium branch with ENA and EK and G leak and E leak and so on. And so, this is our I external, that is the input that is coming in, and this is the inside and this is the outside, and the difference is delta V, the membrane potential. So, remember, this I external is actually the synaptic input that is coming into the soma after traveling through the dendrites, or if the synapse is on the soma itself, it is the output based on the synaptic currents. So, remember that it is essentially an excitatory postsynaptic potential or inhibitory postsynaptic potential, or the corresponding current, inhibitory postsynaptic current or excitatory postsynaptic current, that is being injected into this neuron model. And if there are many synapses, some the net current reaching the soma is what that I external is representing. So, by the time this current reaches this soma, based on the spike produced by stimulus S1 at a particular synapse, or based on the spike produced by stimulus S2 at that same synapse, those currents have to be very different in order to produce a different spiking pattern in the soma that will propagate forward. So, what exactly is coming in? If we remember, at the dendrite, if we say that it is not active, in that case the dendrite is passive, then it is the capacitor and then simply a G leak and E leak. So, it is sort of like a leaky integrated, the model for the leaky integrated fire neuron, and here there will be no spikes, but when the synaptic input comes in, all we will see is basically integration and reaching up to a certain level. And in the postsynaptic response, what we will have that after it increases, there is a decay based on the time constant. So, this is time, and let us say this is the IPSC or EPSC in the other direction, or absolute EPSC, let us say. So, this charging and discharging of the capacitor is dependent on the time constant, is determined by the charging time and discharging time is determined by the capacitance of the membrane and the impedance or resistance in parallel with it. And so, that time constant tau, so let us say this is because the spikes are almost like delta functions, this rise is very quick and this decay is based on the time constant of the membrane, that tau. So, now this tau can be very different across neurons, and in fact as we go, it is one of the functional properties of the neurons, that is, if they are integrating over a very long period of time, then this tau is large. If they are acting as very fast coincidence detector type of neurons, then this tau is extremely small. So, if you think of tau to be nearly 0, that is very close to 0, in that case any difference between the spike trains, any difference in the two spikes, any difference in time between the two spikes, we have taken the simplest case where there is one spike in both. Actually, not the simplest case, there can be no spikes in the other one, that is the trivial case. So, other than that, this is probably the simplest case. And if there is any little time difference between the two spike trains and this tau is extremely small, then the postsynaptic neuron would always be able to discriminate between them. However, if the tau is very large, then the difference in the postsynaptic current is going to be negligible in terms of what reaches the soma. That is, if we think of tau to be 0, the postsynaptic current is simply a blip corresponding to the spikes S1, S2, or postsynaptic potential, this thing. However, if it is so, this is the case for tau equals 0, these two cases, and for tau equals, let us say, going towards infinity, then this is never going to come back down, and so the postsynaptic potential is going to be like this, or current is going to be proportional to like this. So, this is the case where our tau is approaching infinity. So, these are the two extremes. So, in order to study the study the postsynaptic neurons behavior for a range of taus, we can use a discrimination measure that depends on this tau by considering sort of the postsynaptic potential that is created by two spike trains. So, let us say we have this tau that we will be varying from 0 and to a very large value going towards infinity. Let us say, and obviously 0 is also, we won't be able to do it, it is a very small value. So, let us say we have 1 millisecond, 10 millisecond, 100 milliseconds, and what have you, 1000 milliseconds, and so on, and intermediate values. So, by varying tau, we can consider many possible postsynaptic neurons, and only one synapse in this case. So, now if we have R11 or Rij is a spike train and Ri prime j prime is another spike train, keeping our original nomenclature. Let us say it is some spike train like this, and this one is another spike train that is like so. We can think of postsynaptic potential corresponding to each of these spike trains by convolving each of the spike trains with exponential decay, which is the essentially the shape of the EPSP for produced by the spike train, an idealistic shape. So, this is e to the power minus t by tau, and this tau can be varied. So, now this is work that was done and proposed and done by Van Rossum. So, in response to in this spike train Ij and spike train I prime j prime, if I convolve that spike train with this e to the power minus t by tau, let us say we will get some function like this. And similarly, for the other spike train, we will get something like this. Now, for the different taus, these shapes are going to vary. For tau going towards 0, they are going to be very similar to the spike trains with delta functions at each of those points. So, what distance is now being proposed is that, let us say that this postsynaptic potential, proxy for the postsynaptic potential, the Euclidean distance between these two curves. So, after the convolution, so let us say Rij is a spike train which is summation some delta function t minus Tij, and Ri prime j prime is another delta function t minus Ti j prime, a sum of delta functions, saying that those are the spike times. Tij are the spike times in the Ij spike train, and Ti prime j prime are the spike times in the Ij spike train. Actually, there should be the spikes that have to be represented. Ij is representing the spike train, and so there must be, let us say, k and k prime, and so this is summing over k, this is summing over k prime. So let us say this is going from 1 to p, and this is going from 1 to q, this is going from 1 to p. So p spikes in the ij spike train and q spikes in the I prime j prime spike train, and that is being indexed by k. So this needs to be convolved with the e to the power minus t by tau. So we call this as a function ij, which is rij, and convolved with e to the power minus t by tau, can be proportional to that. Similarly, let us say f I prime j prime is proportional to r I prime j prime convolved with e to the power minus t by tau. So this is the white curve that we have here. This is our Fij, and the cyan curve function that we have is our Fi prime j prime over t. And we are saying that the Euclidean distance, that is the integral, let me use the white color here, the integral of Fij minus Fi prime j prime square as a function of t dt, and then normalized by the duration over which this calculation is being done. This is the distance metric. Obviously, we will have to discretize it and then do the calculation, but for analytical purposes, for the cases of the tau equals 0 or tau going to 0, tau going to infinity, we can use this integral form to calculate actual distances. And this distance metric can be very useful, and actually is sort of a biological way to look at distances between two spike trains, although it is not necessary that the Euclidean distance is how the postsynaptic neuron is going to behave, but that is one way to think about it. At least that is what is happening inside the membrane potential, and on top of this there will be other inputs coming in, which is why this is not that straightforward, but it gives us an idea of how much dependence there is in the responses that can be biologically decodable. And so using this d, and for a particular tau, we can quantify the mutual information. So now so what we have is, now we have a d that is a function of tau. So, and based on this d as a function of tau, for the case of each tau, we will have a mutual information between s and s hat that is a function of tau, or we can simply say I tau. And so we can plot this I tau as a function of tau, where this is 0 and on the other end we are going to infinity, and we can get the mutual information for the different taus to be of different shapes, and that the shape of it is instructive in the sense that it tells us how the postsynaptic neuron's properties is going to use the responses and determine the discriminability between stimuli. So now to come with the physical meanings of these. So when tau is 0, as we said, the postsynaptic neuron is almost behaves like a coincidence detector, which means that the postsynaptic neuron is acts so fast, the time constants are so small that it can discriminate between very fine time changes in spike trains in the two cases, two stimuli. That is, or in other words, it can only when the spikes coincide in the two cases can the neuron not be able to discriminate. So, in another example, we will see later on that this coincidence detector is actually can also be thought of in another way, that is, the neuron responds only when it gets coincident inputs from two synapses. It is akin to that, it is only now being applicable to two different stimuli under in the same synapse at two different times, and it is almost like it is using the history to say whether the spikes occurred in the same time or not. So this case is the case where we have extreme fine time resolution. And as the tau increases, this is the case of rate based encoding. Why, we will see in a moment why it is rate based encoding. Now, what we mean here extreme fine time resolution is that okay, the spiking is I mean the discrimination or the decoding is dependent entirely on the spike times, I mean totally on the spike times. Of course, the number of spikes play a role, but it is the spike times that play the most important role. Whereas in the rate based encoding, where the tau goes off towards infinity, the timing of the spikes wouldn't matter. So if we randomize the locations of the spikes, the distances would still be the same. So as we go towards on this side of this graph, we are going towards rate based coding. As we go towards this side of the curve, we are going towards spike time based encoding. So we have spike time, and here we have rate based. So why is it rate based over on that side? So if you do the calculation, where if we take tau goes to infinity, let us say there are p spikes in R11 Rij and q spikes in Ri prime j prime. So let us say these are the p spikes, and in here we have q spikes. So this is stimulus S2, this is stimulus S1, or it doesn't have to be Rij and Ij prime, Ri prime j prime. So if tau is very large, as we were saying, in the case of the Rij, what we will have is jumps of equal amount at each and every spike location, and for the cyan case, again we will have similar jumps at each and every spike location, And so when we look at the difference between the two and integrate over time over the entire duration, square it and integrate over the entire duration, it will turn out to be simply the proportional to the difference in the number of spikes p minus q at the end, the cumulative value at the end, because the difference is going to be at the end go on up to infinity, and the difference is p minus q. The initial differences are going to be negligible till it builds up to p minus q. So that means it is totally rate based code, that is the difference in the number of spikes is what the distance is. Similarly, if it is purely spike time based where tau is 0, if we do the integration again by taking the difference and squaring it, you will see that it is going to be proportional to the sum of the number of spikes or squared of that. So that way it does not matter how many spikes are, the difference in the number of spikes, it is rather just the timing. Each of the spikes contribute equally to the distance. So this is one of the ways in which we can quantify discrimination and quantify dependence between stimulus and response using a biologically plausible mechanism of decoding. So there can be extensions of this idea very much, and essentially it is the profile of this I tau that will be obtained that will tell us a lot about how the discrimination changes based on the postsynaptic neurons property and so on. So there are other measures of discrimination like the Victor and Purpura method, which is very much similar. We will not go into that here. It is essentially a way by which we convert one spike train to the other spike train, and by shifting spikes or adding or deleting spikes, and there is a cost associated with the shifting of spikes or adding and deleting spikes, and that cost is basically like the tau here, or 1 over the cost becomes the tau. We will not go into details here, but just for your sake you should know that there are other measures like this, and they can be used again in the same framework of computing mutual information between stimulus and response based on discrimination and biologically plausible measures of discrimination. So with this we will conclude our lectures on discrimination based understanding of coding and decoding, and in the next lecture we will talk about some more specific ideas of discrimination based understanding of behavior based on some examples. Thank you.